Hello, my name is jchen, and my research project, conducted alongside my partner Tamir Zitelny, was impaired geotaxis in Drosophila using LV-Gal4 UASQ16 and UASQ128 targeted gene expression. Our research was ultimately centered on Huntington's disease. Weiss et al. Characterizes Huntington's disease as a disorder that is characterized by protein aggregation caused by an excessive number of CAG repeats in the Huntington gene. This in turn leads to aggregation of the Huntington protein in the cells of basal ganglia, disrupting vesicle trafficking and protein folding as according to Goentero et al. As Tamir et al. Notes, Drosophila melanogaster can be used as a model system to study the association between the number of polyQ repeats and the onset of impaired locomotion because of its complex neuronal networks. Utilizing the LV-Gal4 UAS system, the expression of either polyQ16 or polyQ128 was driven to expression in neuronal cells exclusively to observe if certain lengths of glutamine repeats can affect neuronal function. The LV-Gal4 UASQ16 and Q128 crosses were carried out as described by Kosinski, Collins et al. The four genotypes utilized to create the 64 progeny in this experiment can be seen here. After the 64 progeny were collected and separated into four groups of 1. Random wild-type Q16, 2. Random wild-type Q128, 3. polyQ16, and 4. polyQ128, all of which are females, a startle-induced negative geotaxis assay was performed to observe whether CAG repeats impaired locomotion of the flies. 8 flies of each genotype were placed into a graduated cylinder that was separated into 3 sections using 2 endpoints. The 4 trials were repeated twice, totaling 8 trials of startle-induced negative geotaxis assays performed. Proper geotaxis was defined as reaching the second endpoint. As seen in this bar graph illustrating the total distribution of Drosophila in the negative geotaxis assays, there was an average of a 76.9% decrease among the number of polyQ16 flies that exhibited proper geotaxis compared to their wild-type Q16 counterparts. Among the polyQ128 flies, there was an 80% decrease in the number of polyQ128 flies that exhibited proper geotaxis compared to their wild-type Q128 counterparts. The 2 pie graphs below illustrate the distribution of flies that exhibited proper geotaxis by reaching the second endpoint in trials 1 and 2. Overall, it was found that the wild-type flies, on average, constituted 82.1% of the flies that displayed proper geotaxis. The polyQ16 flies, on average, constituted only 10.7% of the flies that made the second endpoint. The polyQ128 flies constituted an even lower percentage of flies that displayed proper geotaxis, on average at only 7.1%. Our findings suggest that there is a direct relationship between the development of HD and the presence of CAG repeats in the HTT gene. Thus, it was suggested that expression of both polyQ16 and polyQ128 increased the likelihood of disrupted geotaxis in Drosophila, as evidenced by the 76.9% decrease and 80% decrease in the polyQ16 and polyQ128 flies respectively that were able to reach the second endpoint in time. Thus, there was an association between the presence of polyQ repeats in the HTT gene and impairment of locomotion in Drosophila. Furthermore, the number of polyQ16 and polyQ128 collectively totaled only 17.9% of the flies that reached the second endpoint, further suggesting that polyQ repeat expression was directly contributing to disruption of neuronal motor control. Since the random wild-type flies cumulatively composed of 82.1% of the flies that displayed proper geotaxis, this was suggestive that no expression in CAG repeats did not impair neuronal control of locomotion. Further studies will investigate whether impaired geotaxis of the flies was due to disruption in particular neuronal structures involved in coordination or balance. Future studies will assess which specific neuronal structures in Drosophila are affected by both polyQ16 and polyQ128 expression to evaluate which branches of the nervous system HD mainly affects.